Hi guys, welcome back. Today we're going to be solving week 5 Biology AMS. So let's begin. The first question tells us, what is gene expression? So as we learned before in chapter 10, gene expression is the use of genetic information to produce proteins. All the other options are wrong. Okay, so this is the first part from chapter 11. We have blood groups, okay, and we have to know what are the expected phenotypes. They gave us the parent's genotype, so look what we will do. So first of all, you guys have to know this table, okay? So this table over here on the side about the blood groups, you all must be aware of it. So how do we solve or how do we draw the Punnett square? So since the parent's genotype, I'll start with the female. It's IA and IO. So I'm gonna put IA and IO. And then like a square, I check the man's genotype, IB and IO. So IB and IO. Then I can directly, so IA and IB. So it will be IA, IB. This is gonna be what? AB blood group. IBIO. So IBIO. This will be B blood group. Then I have IAIO and then IOIO. So these four are my genotypes. Okay? So IAIB, since I have four, okay, so the total is 4. For it to be AB, I only have one AB, so 1 over 4. And then again, over for AB is 1 over 4 the probability. 1 over 4, so 25%. Okay? Now for blood group B, I have a total of 4. So always put the total number in the denominator. And I only have one blood group, which is B, so 1 over 4, so it will also be 25%. Now same thing for A. I only have 1 over 4, 25%. And then finally for O, I only have 1, so 1 over 4, so 25%. And always, guys, all the sums of all your probability should always add up to 100. If they add up to less than 100, then you've done something wrong. If they add up to more than 100, then you've also done something wrong. So what are the expected phenotypes? We said 25% group O. Because they asked about the phenotype, we're not going to say IAIO. We directly give, this is the phenotype, okay? And that's it, 25% for O, A, B, and A, B. Okay, this is also a similar one. We have to describe the inheritance of ABO blood groups. So what are we going to do? Again, check the gametes. So the man's gametes are what? Only IB. So I'm only gonna put IB. And then for the female is IA and IO. So IA and IO. And it doesn't matter if you put the female up or the male up, okay? It really doesn't matter. So what are we going to do? IA, IB, and IO, IB. So now I only have 2, so my total is 2. 4. This gives me what blood group? It gives me AB blood group. So this AB is the phenotype, 1 over 2, so that's 50%. And then IOIB gives me the B phenotype, B blood group. Only have 1 over the total is 2, so also 50%. And 50 plus 50 add up to 100. So my answer is correct. So 50% blood group AB and 50% group B. Okay, which of the following are true about sex-linked genes? So before we look at the options, whenever we talk about sex-linked genes, so remember, just a quick reminder, we talked about we have 23 pairs of chromosomes, correct? And we said from these 23 pairs, we have 22 pairs. We call them autosomes. And we have 1 pair, which we call the sex chromosomes. And what is the function of the sex chromosomes? They determine the sex. So for a male, he will have XY, while for a female, she will have XX. Okay? So when we talked about sex-linked genes, they're always found on the... Okay, so sex-linked genes are found here. Okay? On either this or that. Now we have Y-linked and X-linked. But for this exam, and I think for your level, they're just going to be focusing about the X-linked diseases. So when we talk about X-linked diseases, they are found on the X chromosome. Okay, they cannot be found on the Y chromosome. Now, since they're found on the X chromosome, females have two X chromosomes. Okay, so the chance of them getting the disease is not as high. Why? Because we can have, remember, when we talk about alleles, we have a dominant allele and we have a recessive allele. Okay, for the phenotype to show, I only need one dominant allele, while for the recessive phenotype to show, I need two recessive alleles. So for example, if I tell you, usually we denote the dominant allele by a capital letter and the recessive by a small letter. So let's say A is the dominant allele and small a, lowercase, is the recessive allele. So for a person to show the dominant phenotype, he only needs, for example, IA. If he only has one dominant allele and the other allele is recessive, he will still show the dominant phenotype. While if for the recessive phenotype to show, I must have both alleles to be recessive to show the recessive phenotype. So we don't get confused. Let's go back over here. Why are males usually more affected by sex-linked diseases? Because in the female, there is a chance that if I have one allele which is recessive and this causes a disease, I have a chance that the other allele to be dominant and does not cause the disease. So I will have the dominant phenotype and I will not have the disease. I will just be a carrier for the disease. However, for males, since they only have one X, they can, if they get the recessive allele, there's nothing else to mask the effect of the recessive allele as we saw in the female. I hope that makes it clear. So let's look at the options. They are found on either sex chromosome, correct, either the X or the Y. They are identical in males and females, that is incorrect. X-linked genes inherited by males are always of maternal origin. When we say maternal origin, means from the mother. Why is it from the mother? Because remember, to get a son, the dad gives the Y chromosome, while the female gives the X chromosome. So it's always going to be from a maternal origin. And while Y-linked genes are present only in males, that is correct. Y-linked genes are inherited by females. Females do not inherit Y-linked genes because remember, it's XX for females and XY for males. Okay, which of the following accurately outlines the features of X-linked recessive disorders? More females than males are affected. We said that is incorrect. We said more males are affected. An affected son always has a mother who is affected. Okay. An affected son always has a mother who is affected. It depends. Hello, we'll get back to it. So for a female to have the characteristic, her father must have it. That is correct. Okay. The characteristic usually skips a generation from the grandfather to the grandson. Yes, it's because this characteristic can get lost if it's too heterozygous. It's not a dominant, especially if it's a recessive allele, it can die out. So that's why we say it skips a generation. If a father has the trait, all of his sons will have it. Remember, we said that is incorrect. Why is it incorrect? Because the father gives the Y chromosome, okay? Not the X chromosome. The X chromosome comes from the mother. If a mother has the trait, all of her sons will have it. That is correct. Because remember, We said the mother is the one that gives the affected allele. Okay? Okay, now we're going to be talking about a test cross. What is a test cross? So before we look at the options, so usually a test cross is what? Is to determine if the dominant phenotype, so to determine if dominant phenotype is homozygous, so if it's homozygous dominant or heterozygous dominant. Okay? So let me give you an example. Let's say we have a dominant phenotype, okay? Let's say capital A is black fur, small a is brown fur. Now I have, for example, a cat that has black fur. Now the cat could either be AA or one dominant and one recessive, or both dominant. Okay, because remember the phenotype will be the same. Now I want to know if it has this or that. So what do? I will cross it with a homozygous recessive. Okay, it makes sense. Look why it makes sense. So let's try. So if I cross it with a homozygous recessive, if it was heterozygous dominant, so A, and I only have one allele, which is A, I will get these phenotypes. I will get a 1-to-1 ratio. I'll have one dominant and one recessive. So if I get a 1-to-1 ratio, one dominant and one recessive, I'll know that my dominant phenotype was heterozygous. Now, if it was homozygous, what will I get? I will only get one phenotype, which is the dominant phenotype, and it will be, I will directly know that my original phenotype was homozygous dominant. So let's look at the options. It is performed to determine the genotype of an individual with a dominant phenotype. That is correct. It involves crossing the individual with the dominant phenotype with a homozygous recessive, as we said, and a phenotypic ratio of 1-to-1 will be observed if the individual with the dominant phenotype is heterozygous, as we said. If the dominant phenotype was homozygous, what did we say? We'll only get one phenotype, which is the dominant phenotype. It can be performed between individuals of different species. Of course not. We must have the same species because we don't want any other factors to play with it. And a 1-to-2 ratio with the dominant phenotype is homozygous. No, we don't get a 1-to-2 ratio. Okay, which of the following are functions of mitosis? We already spoke about mitosis, so I'm just gonna skip over it. Okay, the picture below shows a rabbit A with black fur. Black fur is a dominant trait. Okay, so directly, guys, on your scratch paper, write black fur is dominant. Okay, and it's gonna be denoted by a capital letter. I think it's probably gonna be capital B. Okay, let B denote the black fur dominant trait, and small b lowercase denote the white fur. So white fur is recessive. Okay, this is the first step you have to do when you solve such questions. What are the possible genotypes for rabbit A? So since rabbit A has black fur, it can either be homozygous dominant or heterozygous dominant. So we're gonna choose 2 capital B's and 1 capital 1 letter. Now, how do I find the genotype of the dominant phenotype? As we just explained before, we perform a test cross. And what do we test cross it with? We test cross it with a homozygous recessive, which is the white fur rabbit. And if I get a 1-to-1 ratio, I directly know that my dominant phenotype was heterozygous dominant, so I will choose capital B and small b. So which of the following accurately outlines the features of X-linked recessive disorders? So more males will be affected, we already spoke about it. An affected son can have parents who have the normal phenotype. So I can show it right here. Let's say the female, let's put it again. Okay, so let's say the female has XA and XA, and the male has XA and Y. So the female and the male have normal phenotype. Now, if they have a son, he's gonna get the Y from his father. Now, there's a chance he gets the good allele from his mother, but there's also a chance he's gonna get the recessive allele from his mother, and here he will be affected. So this shows us that a son can be infected even if his parents have normal phenotypes. The characteristic usually skips a generation. We said it usually skips a generation from the grandfather to the grandson because if it's a recessive allele, it could be lost. Okay, if a woman has the trait, all of her sons will have it. We already said the reason why. Let's say a female has the disease, while, for example, the father does not. Okay, her sons will get the Y from the dad, but always their X is going to come from their mother, maternal origin. So if the mother is infected or affected, her sons will be affected also. And we already said why if a father has a trait, all his sons will have it, why it's wrong. Now, this is an example of a disease which is X-linked. Why is it X-linked? Because it exists on the X chromosome, and it's recessive because it can be masked by the presence of the normal gene. Whenever they say heterozygous, it means they don't have the same allele, okay? Non-affected woman, so of course, is going to have one dominant and one recessive. The possible gametes, it's either going to be the dominant allele or the recessive allele. Of a man, he's going to have the recessive allele since he has the disease, and the gametes would be XA with lowercase and the Y. Now, if heterozygous non-infected woman, so heterozygous non-infected woman, so she's going to be XA and XA, okay? Mates with a hemophilic man, so he has XA and Y. What are the genotypes I will get? I will get XA, 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 XAY, and XAY. Now, if I look at her sons only, so these are her sons, and these are the daughters. If I'm going to look at the sons alone, 50% will be affected, right? Since I have two sons, that's my total. One of them is affected. This is the affected one, so 1/2 is 50%. And then 1/2, 50% unaffected, which is this one. While for the daughters, also have the same case. I have a total of two daughters. The chance of one being unaffected is 1/2, so 50% unaffected, while this is affected, so 1/2, 50% affected. So 50% of the children will be affected. Now, the first one they're asking us all the children, so over 4, and I have two children that are affected, so 2/4, which is 1/2, which is 50%. For the boys alone, we already explained it, why it's 50%, and same thing for the girls alone. Okay, we're gonna skip this. If you haven't watched my video for meiosis and mitosis, make sure to watch it. You'll be able to answer the questions very easily. Also, this is previous material, so I'm just gonna skip it. Also, this one, same thing. Again, those are all easy. If you have questions, let me know in the comments down below. I'll answer all your questions. Plus, there's also videos already about them on my channel. These are all diagrams. You just have to know them, and they're straightforward and easy. Okay? And always make sure you know how to label these questions, because they will ask you to label in the exam most probably. Okay? Okay, let's do this question. So we have a pedigree chart represents the inheritance of color blindness in a family. They're asking us if individuals 3 and 4 have another son, what is the probability that he will be color blind? So 3 and 4 are over here to the left. Now, what we will do is first we're gonna check their offspring. So they get, and always pay attention to the key and understand the key. So I'll have one normal female, which is individual 11, and I'll have one normal male, which is individual 12, and one colorblind male, which is individual 13. Now, what do you guys know about color blindness? Color blindness is a X-linked recessive disease, okay? Is an X-linked recessive disease. And look how we can also know from the pedigree. Before I tell you, think about it very quickly. What did we say about X-linked? Who will be more affected? Males. And if we look at the pedigree, only individual 2, we have a colorblind female, while all the other individuals who are colorblind are males. So we can directly see that males are usually more affected by X-linked diseases. So we assumed and we put that assumption that colorblindness is an X-linked recessive disease. So since an X-linked recessive disease, what are the genotypes of individuals 3 and 4? So for the colorblind male, of course, it's going to be XA and Y. Now for the female, how do I know the genotype of the female individual 4? I'm going to look at her parents. Her parents are individuals 1 and 2. So 1 is a normal male. So individual 1 is a normal male. So he has XA and Y. While individual 2 is a colorblind female. So since she's colorblind, she has 2 recessive alleles. So she can only give X with lowercase a. So what are the genotypes? XA, XA, and XA, and Y. And that's exactly what we got. Individual 3, we have a colorblind male. This is the colorblind male. For individual 4, it's a carrier, X capital A and X small a. So these are the gametes she can give. Now they ask us if they have another son. So instead of doing this and this, I don't care about it because I'm just looking at the sons. So I will get XA Y and X small a Y. This is affected, while this is not affected. So my total is 2. 1 is affected 1 over 2, so 50%. So the answer should be 50%. Here they're just, as I told you guys, always be able to read the key. It's very straightforward. They're asking us how many colorblind males there are. I check the key. Colored square is colorblind male. I count how many colored squares: 3, 13, 7, and 17. So that's 4. And colorblind female, it's a colored circle, only individual 2, so 1. Okay, here they're telling us color blindness is caused by a recessive allele on the X chromosome. It's a sex-linked condition. A normal-sighted female married a color-blind man. So since she's normal-sighted, what percentage of their offspring are expected to be female carriers? And the second question, what percentage of their offspring are expected to be normal-sighted males? And what percentage of their offspring are expected to be colorblind males? So normal-sighted female. So here I'm assuming when they tell us normal-sighted, they mean she is homozygous dominant and not a carrier. If she was a carrier, they should have specified that the female is a carrier. And we have a normal-sighted male. I don't need to write XA twice because she's only going to give one gamete, which is XA. So I'll get XA, XA, and XA, Y. Oh no, sorry. She marries a colorblind male. So it's gonna be lowercase, lowercase, and lowercase. So what do I get? I get a female carrier, and I get a male who's, wait, I messed it up. So XA from the female, and we have lower letter and the Y. That's the male because the male is colorblind. So I will get XA and XA. This is a female who is a carrier, and I'll get XAY. This is a male who is unaffected. So if I look at my options, to be female carriers, 50%. Because here they're asking us about the offspring, so it's always going to be over 2. Female carrier, there's 1, so 50%. Same thing, normal-sighted males, we have 2 because they said what percentage of their offspring. So they didn't specify if male or female. 1 over 2, so 50%. What percentage is expected to be colorblind? This is 0 because we didn't get any colorblind males. Okay, sex-linked recessive alleles. Of course, the males inherit 1 copy because their sex chromosomes are X and Y. If males inherit, okay, so since they told us the effect is seen immediately, the males only need to inherit 1. However, if females inherit 1 recessive allele, then its effect can be masked by the second allele on the second X chromosome if it is dominant, as we explained before. So females inherit 2 copies of the genes that are on the X chromosome since their sex chromosome is XX. A pedigree is a chart that shows the ancestral history, okay? The following pedigree illustrates the inheritance of fur color among a group of related individuals. Are each of the individuals 1 and 2 in the pedigree shown most probably heterozygous or homozygous? How am I gonna know? Okay, since they show the phenotype, they are dominant. But in the offspring, I have 1 that is recessive. So if the parents were both dominant, so homozygous dominant, there is no chance for me to get a recessive. Okay, so recessive, there is no chance. So of course they have to be heterozygous to be able to get individual 6 genotype. Okay, a woman having red-green color blindness and a male with a normal vision have a child. Let X denote the normal allele and X lowercase to denote the color blindness. So what do we know about here? It's very straightforward. They give us the phenotype, the parent's genotype. Since a healthy man, it will have the dominant allele. Since it's a colorblind female, she will have both recessive alleles. If their child was a girl, what would her genotype be? So since she's a girl, she's gonna get the X from her father, which is a capital B, so X capital B, and she will get another X from her female, from her mom, and the mom can only give X lowercase b, so this is going to be the genotype of their daughter. Okay, here they're telling us the black fur is dominant over the white fur, so of course we're gonna denote the black with a capital while the white with a small letter. It's a cross between 2 heterozygous individuals. So just a small trick for you guys. Anytime you have a cross between 2 heterozygous, okay, the ratio will always be 3:1. 3 dominant phenotype, this is the phenotype ratio, okay, and 1 recessive phenotype. While for the genotype ratio, so this is the phenotype ratio. For the genotype ratio, I'm gonna show it to you, and then instead of wasting your time in the exam, you can directly put it as it is. So for the genotype ratio, it's going to be 1 to 2 to 1. This is going to be homozygous dominant, while this will be heterozygous dominant, and this will be homozygous recessive. Look why. So since I have two heterozygous, so A, and A, I'll get AA, and AA. So look at my genotype. Or first, we'll start with the phenotypic ratio. Dominant phenotype, dominant phenotype, dominant phenotype. So 3 dominant phenotypes. While I only have 1 recessive phenotype, so this is why the phenotypic ratio is 3:1. Now for the genotype ratio, I have 1 homozygous dominant, I have 2 heterozygous dominant, and I have only 1 homozygous recessive. And this is just an example of they showed how we did it. Okay, the allele R that codes for red flowers is dominant over the small r that codes for white flowers. 2 red flowers were crossed. We had a ratio of 90 red flowers and 30 white flowers were obtained. So I can even not solve it directly from the ratio, since it's 90 to 30. 90 to 30 is what I'll get 3:1. And if this is my phenotypic ratio, I'll directly know what it was a cross between 2 heterozygous individuals. So what are the genotypes of the parents? Of course, it's going to be, both of them will be heterozygous. What is the probability of having offspring with RR genotype? So now we look at the genotypic ratio. It's heterozygous, it's 2, so it's going to be 50%. Okay? Then homozygous dominant, there's only 1, so it's going to be 0.25%. Okay, or 0.25. And also homozygous recessive would have been 25%. And if we sum them all up, 25 plus 25 plus 50, I will get 100%. Okay, let capital N denote the normal allele and small l denote the allele responsible for the recessive condition. So, both parents, they're asking for the genotype. So since they have a member who has the disease, the parents should be heterozygous. So that's why you put heterozygous. Now, for their offspring, individual 1, since has the disease, of course it needs both recessive alleles. That's why it's small letter n, small letter n. Now, for individual 2, we cannot tell. It can either have a dominant homozygous or a heterozygous dominant. Okay, this is easy also. So how first you should know how to differentiate between male or female. Of course, the first one is male because this is the X and then this is the Y. Since it has a small letter, then it is affected. Second one is a female, which is a carrier, since it has the small letter 2. Third one, it's a female, which is unaffected, so it's homozygous, healthy female. Fourth one would be a healthy male, and the last one would be a female, which is affected. Okay, here they told us we are crossing a heterozygous male animal with black fur, so capital B, small b, with a homozygous female with white fur. So it must have both recessive alleles. So that's why we put capital B, small b, small b, small b. The gametes, it's easy to tell. And then the offspring genotype. So let's do them. So these are the male gametes, while these is the female gametes. So this is the result I will get. I have a dominant phenotype, and I have a recessive phenotype. So my genotype will be 50% of this and 50% of the other one. The phenotype, 50% black fur, 50% white fur. which allele for body color is dominant, we can tell from the results they gave us. So when we crossed fly A and fly B, they were all gray. I crossed fly A and fly C, they were also all gray. When I crossed also fly B and fly B, I get 3 to 1. Remember the 3 is usually the dominant, so gray, I can directly tell that the dominant color is gray. Which group of flies is heterozygous for body color? How did I know it's B? Because remember, when we crossed 2 heterozygous, what was the ratio we got? 3 to 1. Here I also got a ratio of 3 to 1, so fly B is heterozygous. Which group of flies is black? It's going to be C, because we said flies B are heterozygous, so they have the dominant allele, so they will have the dominant phenotype. If flies of group A were interbred, okay, so if we basically made flies from group A, since group A is dominant, of course they will all be gray. And which two groups have the same phenotype but different genotypes, A and B? Because we said B has a heterozygous genotype while A will have a homozygous phenotype. Okay, this is the same thing as the question we did before. Okay, so I'm just gonna skip over it. Okay, so if a homozygous non-affected woman mates with a hemophilic man, what are the expected phenotypes of the children? So, since the female is unaffected, so it can only give the good allele, and the male is affected, so he has the recessive allele and the Y. So XA, and XAY. 0% of the children will be affected. The female will be a carrier, but not affected, remember, and the male will also be unaffected. So 0% will be affected, 0% of the boys will be affected, 0% of the girls will be affected. Okay, whenever they tell us the genetic makeup, it's always going to be the genotype. If they ask you about what you see the phenotype. If I look at someone with, for example, orange hair, I'm not going to know his genotype, but I can directly tell his phenotype is that he has ginger hair. Okay, these are two important definitions you have to know. When you have different alleles expressed at the same time, it's codominance, and an example would be the AB blood group, like we see in option C, while incomplete dominance is when you have an intermediate. So whenever you see an intermediate, it's when the dominant allele does not completely mask the recessive allele, so they go for an intermediate in the middle. Okay, and they carry identical alleles, homozygous, different alleles, heterozygous. Okay, I will explain this question. So remember during meiosis, why did we choose this one? So they're asking us about the mass of DNA. So before meiosis happens, what do we have? We have DNA replication. So that's why the mass of DNA increased. And then we have division, the first division of meiosis. But here we're still haploid, okay? I mean, we're still diploid. And then in the second division, we become haploid, so that's why the mass of DNA again decreases. Okay, this is also easy. This is also straightforward. Even this. Okay, we already spoke about test cross. We said we cross an individual having the dominant phenotype with someone who is homozygous recessive, and if we get a 1-to-1 ratio, then the dominant phenotype had which genotype? It had the heterozygous genotype. And that's it for today's video. I hope you all understood the quiz, and I tried to make it as clear as possible. If you have any questions, let me know in the comments down below. And finally, don't forget to like, comment, and subscribe, and share the videos with your friends so we all can benefit. Thank you all, and best of luck on your exam.